Hey, hello Amid, hello Hassan. Nice to welcome you this morning to the Powlett Lab. We are located in the SP building at Loyola campus. This is a biochemistry lab. My name is Sylvie, I'm a PhD student, and our lab does research focusing on mechanisms of iron acquisition in bacteria such as E. coli. Bacteria need iron to grow, and in order to capture the iron, to scavenge iron from their surrounding, they have to make compounds that are called siderophores to secrete them to bind the iron for them to acquire. Now, the focus of my research is on how synthesis of enterobactin occurs, and this is done through concerted action of several proteins, enzymes that are proteins, and we'd like to see whether, I'm investigating whether these proteins assemble in a supramolecular assembly, whether they interact closely. For instance, we have several proteins involved in the synthesis of enterobactin. My goal is to determine whether they are cooperating in a matter of speaking to facilitate the flux of this production. So naturally, the best way to do this is to extract protein and to study them in vitro or in vivo. And a lot of my work involves protein purification and protein separation, which is what I'm presenting to you today. This is a major part of my work, and I just want to give you a brief summary of what this entails from the raw material to the finished product. So the beauty of working with bacteria is that you can tweak them to your liking, and you can use them literally as factories. Bacteria such as E. coli are very good at uptaking DNA from outside, and we can create stretches of DNA, which are called plasmids. And this is engineered to our specification. We can get the bacteria to uptake this, and this is something that we can control for production of the protein through induction of the gene. That I can explain it a little bit later. So it all starts with growth media into which we inoculate the bacteria once they've been tailored to produce the protein that we want. So through incubation, bacteria will grow to a certain level, and then we basically centrifuge the media to create what's called a bacterial pellet. So the button that you see here, this is all the bacteria that are bunched together. They have been producing the protein that I'm after. So my goal now is to extract the protein from the pellet. The way we do this is through cell lysis, meaning I will add in chemicals that will literally burst open the bacteria and release the content, which is a rather slimy goo, which we call a cell lysate. This is very crude. This contains all the proteins that the bacteria has produced, including the one that I'm after. So in order to validate whether my protein of interest is in this media, I have to run my protein, I have to run a sample of my protein through gel electrophoresis. So this basically sends a current down a polyacrylamide gel, and the current will drag the protein according to its molecular weight. And upon staining of this gel, I will be able to determine whether my protein, the one I'm after at this molecular weight, is there. So once that is complete, I know my protein is there. I now need to have it purified, to have it on its own. So this is done through a purification column, where I just pour in the supernatant that I showed you earlier, and through several washes and elution, in the end, I will end up with my purified protein. Now, of course, from the flask over there to this final product, that's not a lot of protein. If I want a larger volume, then I can just scale this up 100-fold by culturing my protein, my bacteria in a larger amount, which will generate a larger pellet. And rather than me manually going over the column, adding the reagents and adding the supernatant, we have automated systems that will do this for me, and in the end yield a larger volume of the purified protein. So this in a nutshell is it. Are there any questions?
Yeah, Sylvie, I guess there's hello. I guess there's no difference between the working principle of automated and unautomated methods. does automated way can offer something more than purification of larger quantity? I mean, can we improve the precision of purification with the automated method? Or
yes and no. It basically speeds up the process. It will also yield much larger volumes. What is different is that when I purify protein in a small column, I have to take several aliquots and measure them individually to see through fluorescence whether my protein is present in which aliquots. Whereas the system that's automated has its own detector, so it will know when my protein of interest passes through which you can see here, through the lens. The computer will know this is the fraction we need to salvage, and it will mark it accordingly in the system. So you have to keep an eye out for it, but you can basically just program it, press a button, and it will do the work for you. Hello, Hassan.
How are you?
I'm very good. You?
Thank you. Actually, do you need to introduce myself?
Go ahead.
Actually, my name is Ehsan Yazdanpanah Muqaddam. I'm a PhD candidate, mechanical engineering. So, first, let's start from this question. How do you use gene expression in your experiments?
As I was saying earlier, you insert a plasmid into the bacteria, and when you engineer this plasmid, there's a sequence there which is called the promoter sequence. This is a stretch that, upon activation by a compound called IPTG, will basically start the transcription. So you can tweak it. You decide for yourself, okay, I have a large enough volume of bacteria. Now I need them to start inducing the protein. So you just basically add in some IPTG. This is very similar to lactose in the way it's working. It's based on the lac operon, which is not something I want to get into right now. So basically you add in this compound in the growth media, the bacteria will uptake it, and it will act on the stretch of DNA, the promoter that's on the plasmid, to induce production of the protein.
Hello, my name is Hamid. I'm a PhD student at Concordia University Mechanical Engineering Department. I have a question about the device you're using, I mean, Western blot. As an engineer, I always interested to know the working principle of equipments. Could you give me some more details about the Western blot with a simple analogy, maybe?
No, this is not Western blot. This is SDS page, gel electrophoresis. Okay. Polyacrylamide gel electrophoresis is a method that's used to separate proteins. So we basically make a gel that creates a polyacrylamide mesh. It's very much like a sieve, and the proteins being coated with a negative charge, upon insertion of the gel into a chamber through which we run an electrical current, the proteins will migrate towards the anode, being that they're negatively charged, and through the mesh of acrylamide, the proteins that are smaller, of course, will migrate faster. And in the end, that's how you end up with separation. I can also show you, there's one I made earlier. We can buy them ready-made, or we can produce them ourselves. It looks like this. It creates little wells into which you insert a sample, and upon inserting this in the cassette, I'm going to do this very, very roughly, of course. Then the electrical current will cause the proteins to migrate faster if they have a lower molecular weight. And after about an hour, it takes about an hour at 200 volts to get a nice separation of your proteins.
Actually, Sylvie, when you talk about your culturing bacteria or growing bacteria, I think the color media you're using is yellow. But the cell media is like pink, and I think so you culture your bacteria in FBS, yeah, or like in cell media.
No, not at all. What you're referring as pink is mostly for tissue culture, for mammalian cells, for instance. For bacteria, we use something that's called LB broth. It's a rich broth that basically contains. It's made with tryptone and yeast extract. It's very rich, it contains all the amino acids, vitamins, and minerals that the cells need. So we make this ourselves, and then you basically just inoculate your bacteria, leave it to grow. Now there's a bit of condensation, unfortunately. You can see that there's some turbidity in there, which is indicative of bacterial growth. And this being rich media, the cell grow rather fast. The doubling time is about every half hour. For E. coli, some cells are a little slower, some cells are a little faster.
When you're culturing bacteria, you're gonna incubate an incubator. Yeah.
We grow bacteria in an incubator shaker. We have one over here. We have other ones in a common room, a common equipment room, but we have our own shaker incubator that we use here in the lab. So you can just, let's pretend it's working. So I grow my bacteria at 37 degrees. It will take a few hours to get to the level that I want. Sorry about this. This is the fume hood going. So once the bacteria has started to grow, I can stop it. I can literally take the flask from 37 degrees, put it in the freezer with glycerol, and then revive them. Bacteria are very sturdy, so I can leave them on my bench, put them back in the incubator. They will start growing again. They will just go quiescent. They won't die. So the best way to inactivate bacteria is really through heat, but room temperature, 4 degrees, minus 40, minus 80. We even have bacteria stocks at minus 80 in glycerol, of course, to preserve them, but you can get them out of there, inoculate them in growth media, start incubating them again, and they will go back.
When I start my experiment, when the cells get confluent, or let's say the cells cover all the substrate, and so, because like me, confluency, it's like a sign for me to start my experiment.
When are my bacteria ready? That's what you want to know. How do I know my bacteria are ready? With bacteria, under constant conditions, bacteria have a growth curve. This is particular to the conditions depending on temperature and the medium to which they're using and to which they're inoculated. But on the whole, it starts with what we call a lag phase, meaning their doubling time and their slow number means you don't really detect them. And then at some point, it gets into an exponential or a log phase where they grow rather fast. They hit stationary phase when the nutrients become depleted, and of course they stop dividing, and afterwards they just die, which is the decline phase. So the best time to use bacteria to induce protein production is when they hit about halfway through the log phase. And how this is measured is through optical density. I will take an aliquot of my culture, put it in a small cuvette, and read it at an optical density, read its optical density at a wavelength of 600 nanometers. So when my optical density at 600 nanometers reads around 0.6 or 0.7, this is the sweet spot telling me that the cells are rapidly dividing and metabolically active, and that's the best time to work with them. It's called the log phase, halfway through. It's analogous to your confluency percentage in tissue culture.
And for last question, so what's the novelty in your project? It's like,
What I was saying earlier is that we hypothesized that the proteins involved in the synthesis of enterobactin, that's the iron chelator, we hypothesized that they interact to form a supramolecular assembly. Now, we know that a lot of processes, a lot of metabolic processes rely on this configuration for a variety of processes. For instance, the Krebs cycle, towards which, with which cells will create energy out of glucose. But there's never been a demonstration that the biosynthesis of enterobactin followed along the same process. And of course, this is very much like in a factory, you see an assembly line where everything is streamlined so that the product of one action will become the substrate of another one. So by determining whether this happens and how the proteins interact and at which point, you have what we call hotspots, the exact interface where the two proteins meet. And once we're able to determine that there is an interaction and where this interaction is taking place, then through drug development, through pharmaceutical industry, you can devise compounds that will basically impede this process and literally stop the bacteria from producing enterobactin and stop them from developing and ultimately putting an end to infection.
Thank you and good luck for your research.
Thank you.
Thanks, Sylvie.